Let us begin with the epidemiology. Malaria, which derives its name from the Italian mala aria, or, quote, bad air, is a huge disease burden. 3 billion people live in endemic areas and are at risk of infection. 500 million clinical cases are seen annually, and up to 3 million people, mostly African infants, die of malaria annually. The red color on this map shows areas at risk for malaria. As is clearly demonstrated, the disease is widely distributed, covering almost the entire African continent, most of South America, and a significant part of Asia. The malaria parasite is Plasmodium, a protozoa. 4 major types of Plasmodium causing human malaria have been known for years. They are P. falciparum, P. vivax, P. ovale, and P. malariae. P. knowlesi was recognized recently as a 5th type causing malaria in humans. Specific vectors for the malaria parasite are about 40 species of the genus Anopheles mosquitoes. Only females transmit the disease because they need a blood meal for the maturation of their eggs. Male Anopheles mosquitoes eat nectar and cause no harm to humans. As a blood contact disease, malaria can also be transmitted through routes such as blood transfusions and dirty syringes, although these cases are rare. Vertical transmission is also possible. The complicated life cycle of malaria includes 3 interconnected cycles of reproduction, 2 in humans representing asexual reproduction with 1 stage in the liver and the other in the blood, and a 3rd cycle in mosquitoes representing sexual reproduction. These cycles are labeled A, B, and C in this diagram. It is important to keep this big picture in mind before beginning to learn about each individual stage. At step 1 in the diagram, the mosquito takes a blood meal and injects sporozoites into the human bloodstream. The sporozoites reach the liver and start reproducing asexually in the process known as schizogony, represented as the exoaerocytic cycle A on the diagram. This stage is asymptomatic. Some Plasmodium species develop brady or hypnozoites or sleeping stages that stay in the liver and can start developing 6 to 13 months later, resulting in late malaria relapses. After reproducing in the liver, parasite reproduction moves to the red blood cells, represented as the erythrocytic cycle B in the diagram. This red blood cell stage gives all of the clinical symptoms of malaria. Here, tissue merozoites, the motile, highly infective form of the parasites, invade red cells and start reproducing in the red blood cells. They develop into immature trophozoites, seen in the ring form under the microscope, which in turn take one of 2 pathways. Some continue reproducing asexually, engaging in schizogony, forming multicellular schizonts and eventually new merozoites that invade new red cells and continue the cycle. Others form new male and female gametocytes or sexual cells, which wait for the Anopheles mosquito bite. At step 8 in the diagram, the female mosquito takes a blood meal and ingests gametocytes, the sexual cells of the parasite. These gametocytes meet within the mosquito gut and start sexual reproduction, known as the sporogonic cycle, C on the diagram, eventually producing sporozoites. These mature sporozoites migrate to mosquito salivary glands. When the mosquito bites a human, they are injected into the human's blood and the cycle begins anew.